Hi, everyone. I hope you're enjoying your first day of e-learning. And I am going to sit in here and I'm going to do a little lesson on sort of connecting some of these ed puzzles that you've had with learning about the cell division, which is kind of what we just ended with. I know you got a lot of new material with looking at the structure of DNA at the end of last week, and I just want to try to help connect the dots for some of you. I'll talk about this also on the Google Hangout tomorrow. But let's see if this works. Okay, so I don't want to just call this slideshow mutations. I'm really trying to show you the connection between what else we've been learning. It's not just about cell division. It's not just about mitosis or something we're going to find out about meiosis, but it's also connecting mutations to that because mutations bring in variation to a population. So it's really important in biology. So just a quick recap here of the cell cycle. We have our different stages. We have G1, S, G2, and M. Remember, M stands for mitosis. This is where cell division occurs at. And when we're talking about mitosis, and I just realized I think I have an error later on, but I'll address it while we're talking. We're talking about somatic body cells. Somatic cells are body cells. So like mitosis has toe in it, and the toe is part of our body. And so that's what we're really looking at when we're talking about the cell cycle, because we need to undergo the cell cycle to divide, to repair wounded cells, and also to grow in number. Before that can happen, though, as a reminder, that the stage between the divisions is called interphase, and the S stands for synthesis, where we're going to be synthesizing DNA. The reason is somatic cells, every body cell, I'm going to keep saying this term so you're used to it, which is a somatic cell. And forgive the quick grammar, I was just trying to put this together, and my spelling's not the best, but it has 46 chromosomes. We get two sets of 23 from mom and dad. So 23 of your chromosomes come from mom, 23 from dad. They might have different genes, and I'm going to have another little session on the relationship between DNA, genes, chromatin, and chromosomes, because that kind of gets a little hazy, but I want to get some props out for that one. But every somatic cell has 46 chromosomes. Oopsie. During the synthesis phase, we need to duplicate it. We talked about how if we don't duplicate it, when we go to divide, then each resulting cell would only have half of the number. And for mitosis, we really want to make identical clones of the original cell. That's the goal here. We want to make additional skin cells if it's a skin cell that's dividing. We want to make additional digestive lining cells if that's what's dividing. Whatever the type of cell it is, you want to make that exact copy with the same instructions. And so before we can divide, we must replicate our DNA. We must synthesize new DNA that's the exact same, making exact copies of the instructions. And let me see if I can move this up. This way, when they divide, they'll each have 46. So if you start with 46 and you replicate, you should have 92 chromosomes in that nucleus. And then when you divide, you'll have half of that number. You'll have 46 on each of the two daughter cells. The resulting cells are called daughter cells. And so just as a review, here's mitosis at a glance. And we're only showing it with 2 chromosomes here, simply to make it a little bit less congested in the diagram here. But you can see that when they replicate, you'll now have 2 copies. And again, there's going to be a future little session on chromosome and chromosome structure and why it forms this little X. But this just means that they're connected here. Same thing with the blue one. We have one more here. It was replicated. So now when we line up here in the middle and we start pulling apart, we're going to pull a red one and a blue one to each side. And you're left with 2 diploid cells. Okay, 2 diploid cells. You can see the other one. If I move it over, I should not have done a webcam. I was trying to be more social. So here is your identical daughter cells to the parent cell, and they're diploid, meaning that they have two copies. They have two sets of chromosomes here. However, sometimes errors occur. And when we have errors in our genetic code, we call that mutations. Sometimes they're helpful, sometimes they're harmful, sometimes there's no effect. Again, my grammar, I should have went back and proofread it before I posted this, but I just wanted to get it up to you quickly. There are different types of mutations. Some mutations happen later on and in certain cells that we're going to talk about in a minute called gametes, and some of them happen in somatic cells. If a mutation is recognized, however, during a checkpoint of the cell cycle, especially right after the replication occurs, okay, and the bases come in and they try to match up, we have these enzymes, which are a type of protein that will try to fix the base pair matchup, so there's no errors in it. We call that DNA polymerase. I'm not going to require you to memorize the name of all the parts that you're going to see on this next slide here, but DNA polymerase is what brings in the bases and binds it to the actual DNA strand, the template strand. And I wanted to talk about mutations now instead of waiting, simply because one of the big times for mutations to occur is actually during that synthesis phase when the DNA is replicating. And so you can see, like, this is kind of complicated looking. We have our chromosome. If we unwind it and take a closer look, we have our double-sided ladder, the double helix structure that you guys made last week. We have an enzyme that helps break it apart. We have DNA polymerase, which comes in and starts binding them here. And so it's kind of a complicated process, but you guys have been working on the POGL, and you can get additional DNA replication details by working through that. So as I said, one of the reasons I wanted to talk to you about mutations, right, when we're talking about the cell cycle, is that there are different types of mutations. One type is a gene mutation. A gene mutation just involves one gene, one particular code for a trait. There's point mutations and frame shift mutations. You can substitute in bases take out the A and you can put a G there. You can insert by mistake an extra base, or you could delete a base entirely. And that does happen. You do not need to memorize the different types, but I just want you to be able to see that errors do occur. And again, I'm going to reiterate, it's not a bad thing. Gene mutations often occur during the cell cycle around when it's being synthesized. And so luckily, if a mutation occurs in these somatic or body cells, it may not necessarily be passed on to the offspring, because we read about how we have these checkpoints in our cell cycle. And if this error, this mutation is recognized, it will send it hopefully into apoptosis. That's not always the case. Sometimes there's an issue. There's an actual mutation with our checkpoints, and we'll talk about that in a minute. It's going to be a big focus of where the next couple of weeks are heading. So the cell cycle in mitosis, as we've discussed it up to this point, deals with somatic cells. Now, there's another type of mutation that can occur. I don't want to say another type of mutation. They're both the same type. They're both errors, but it's what type of cells it occurs in. So we haven't talked about something called meiosis yet, but if a mutation occurs when forming gametes, those are your eggs and the sperm generated through a process called meiosis, because it's in the gamete, it could be passed on to offspring. This is what commonly leads to genetically inherited diseases, and most often those are going to be chromosomal mutations. So meiosis at a glance, just to give you some background, we'll come back to this. It involves, instead of one round of PMAT going through mitosis once, it involves two, which halves the number of chromosomes. This allows for gametes to have half of the genetic info. And if you think about it, that would make sense, because if you had two cells that came together, an egg with 46 chromosomes and a sperm with 46 chromosomes, you'd be left with a fertilized individual having 92 chromosomes. And then when you replicated that for mitosis, you are looking up 184. So you're going to be increasing in number each time. So meiosis is what allows for gametes to have half of the genetic info, and we're going to find out it also has a couple of different processes like crossing over that allows for variation to occur. And when this variation, these spots for variation arise, then there's more of a chance of an error. So really quickly, just at a glance, again, we're going to come back to this. We have our parent cells and replication. Now, if it's a body cell, it's going to go into mitosis. And if it is a cell that produces gametes, it's going to go into meiosis. And you're going to notice that it now has half of the number of chromosomes. Here, they're identical in mitosis, and here, they have half the number they started with. Pardon me. So sometimes when we're looking at meiosis and we're looking at the chromosomes and they come back together here and they're undergoing this first round of meiosis, the first time they go through PMAT, we'll notice that sometimes because they're really close together, things can happen. Like we can have a translocation where they actually cross over, or we can have an inversion where things switch around. And so oftentimes chromosomal mutations, not all the time, there's some definitely genetic ones that occur that are dangerous and that are destructive, but oftentimes chromosomal mutations are severe enough that if something happens, oftentimes the egg doesn't even make it to conception, or not the egg, I'm sorry, the blastocyst doesn't even make it to conception. And I will tell you, I don't think it's on here, just so you guys know, is I actually had a chromosomal mutation. We had a daughter that had an extra copy of a chromosome 18, and she did not make it to she was conceived. I'm sorry, I said conception earlier. But she didn't make it to her birth. And that's just because it's the body's way of trying to weed out dangerous mutations that occur. Other environmental effects are mutagens. It's a chemical or physical agent that changes DNA. Could be radiation, chemicals, or infectious agents here. Here's bacteria spread through contaminated food. If it ends up in a somatic cell, again, it can be passed on to the offspring. Now, some beneficial mutations that occur include lactose tolerance in humans. We're the only mammals that can tolerate milk beyond infant stage. And one of the reasons that scientists suspect for this is changes in our lifestyle when we went to being hunters and gatherers to growing our own crops. Ignore that phone there. I'm going to let it go. And those with this mutation, this error, tended to tolerate the milk from the domesticated cows and domesticated goat and other animals a little bit better than those that did not. On the flip side, negative mutation example would be cancer. When some of these checkpoints aren't working, there's been an error in the checkpoints. Other ones include some more drastic looking ones just by appearance alone. We have our two-headed little turtle here. We have our camouflaged frog here. And then this one, just the pigment color, it's neither beneficial nor harmful. So it can be any of those three. So hopefully this helps you at least get started and gives you a frame of reference. I'll break down meiosis a little bit more. I'll break down chromosome structure a little bit more for you too, just so you can get that terminology down and you can see the relationship. If you have questions, I will be doing a Google Hangout tomorrow, Thursday the 19th at 1 p.m. I'm going to exit and move this down so I can close it out. If I can get this down.